Great. So it's wonderful to be here, and as was said, right, so I'm part of an Allen Discovery Center that's run through Stanford and the University of Virginia. So actually, I think everybody, all the PIs from the center are here. Shane Pierce-Kotler, Casey Huang, Denise Monak, and Jerry Morrison, and even a lot of several members from our labs who all would be thrilled to talk to you. Today I'm going to focus specifically on an aspect of the modeling that we do, and I wanted to start out with a question that I hope will be a little bit provocative, and I guess I have to click something here to get, and we are, or I can use this too, right? Okay, great. So the question is, if you had all the resources you needed to generate all the data that you could ever want, right? I just want you to think about what that would mean, right? What would that data look like? What would it be, right? What would you have when all that was done together? If you were to take all of those resources and generate all of that data, what would actually happen next? Right? What's your end game? You know, Delbruck used to tell his students that they weren't allowed to do a single experiment until they could explain to him all of the possible outcomes and what those outcomes would mean, right? We've come a long way, okay? What would happen next? Somehow we have to figure out what all of that is going to represent in terms of our knowledge. And I think probably everyone here would agree that they would still consider their system of interest far from understood or specified or solved, even if they had all of that data. So we're going to talk about that. How do you get insight? How do you get knowledge from data? And the way that we do it, so our lab is best known for having reported the first construction of the first model of a cell that took all of the genes into account. Okay, so this was for the simplest bacterium, 525 genes. And we were able to use this model to make predictions about how the genetics and the environment came together to produce phenotype. And that was really exciting for us. You know, I think it received some fanfare, which we were thrilled about. But more excitingly to me, in some ways, was what happened next when we started using that model to predict the outcomes of experiments. And there's one that I just want to highlight here. Because what we did is we took a library of knockouts in this organism, and we used the growth phenotypes of those knockouts to predict detailed molecular parameters. In particular, what I think is really interesting is that we were able to use one data type that's easy to generate, which was the growth rates of these knockout strains. And the model kind of gave us this insight that from certain of those growth phenotypes, we could predict a detailed kinetic rate for certain enzymes. And in every case that we tried, granted only 3, very small organism, but in all the cases that we tried, we ended up having very precise predictions of what those parameters would be. And so I want you to hold on to that concept that the data, because it's all connected, right? You wouldn't necessarily think that this growth phenotype is going to give you this other parameter, but because the data is all connected in a real cell, right, you can use it to gain information in a really strong way, provided that you have a mechanistic basis to do so. Okay. Encouraged by those results, we really wanted to push this to the next level. And in thinking about that, we came upon this article, a monograph, kind of a call to action from Francis Crick, where he called for, in the 70s, right, the complete solution of E. coli. Okay, this idea that let's go after this one model organism and try to solve it completely, know everything about it. And so he suggested setting up some kind of a central laboratory which would gather resources, put materials together, distribute, results, a systematic approach where different labs would coordinate, a very bold and provocative proposal that, of course, you probably realize we didn't follow at all, right? We didn't do anything he said, basically. But what did happen was, over these decades, many, many labs continued to study E. coli in their own way, right? And they generated tons and tons of data, okay? So in many ways, this is the closest to the hypothetical I raised at the beginning of the talk, right? If you had all of the data you could imagine in this organism, so you just imagine all of this heterogeneous, very extensive data, what do you actually know, right? Put in Crick's terms, like, do we actually have, or how close are we to a complete solution of E. coli? So inspired by that, we wanted to go after, like, our next target for whole cell modeling has been E. coli. And the first question that we want to ask, beyond just a complete solution, is how consistent the data are themselves, right? This sounds like a simplistic type of a question to ask, but actually it's seen, like, in several fields. Psychology is one, and cancer biology is another. There have been these large meta-analyses which strongly suggest that reproducibility is a problem in the sciences, enough so that Nature put this whole thing together about the reproducibility crisis. So it's actually a fundamental question that we need to ask, like, are these data reproducible, or in our case more deeply, like, are these data self-consistent? Do they tell a consistent, coherent story about what E. coli is? The only way you can ask a question like that or answer a question like that is to figure out a way to integrate and simultaneously evaluate that data in its totality. And that's really what whole cell modeling attempts to do, right? When we go after trying to figure out the completeness of this solution, what we're really starting with is going through and finding all of the papers that have ever been generated in E. coli, right? Gathering and harvesting that data, you know, from all those years, hundreds of labs, millions of data points. And this is all real specs from what we set many of the papers that we pulled together for this. Now, how do you take all of those data? They're heterogeneous. You know, some are large, some are small. They're all over different areas. How do you pull that together? The answer is in equations, right? So you can take all of the equations for every RNA in a cell, right? 4,500 ordinary differential equations, and that pulls together a huge amount of that data. You can write another several thousand equations for the metabolic processes in that cell. Again, it pulls these things together. Same thing with proteins, same thing with many things that I don't have the time to show here. But you can imagine then having all of these equations encapsulating the data, but also encapsulating function. And so you can sort them into their different cellular processes, right? You can build a mechanistic basis for how they all interact and write out then a unified mathematical model that both encapsulates the data and explicitly represents the functional basis behind it. Okay, that's what we're talking about. And once you have something like this, and I should say here, we don't have a whole cell E. coli model yet. We have something more like a half cell E. coli model, not just one half of the cell, but like half of the processes in the cell. And you can but it really does represent a major technical advance for us because it's 10 times the genes, right? 100 times the molecules, and many more different environments. And so you take this and you put it together. We've now got something that can generate, and this is real simulation data that I'm showing you here. You can show how E. coli grows out and divides over time. Here you're just looking at the cell mass as it grows out and divides, right? Here you can see the DNA chromosome as it replicates and divides. Some of the things that we're proud of here, we can now have these simulations be stable for many generations, which turned out to be a pretty difficult challenge. We can also represent more than one environment, which was what the first model could take care of. So if you think about all of these things now coming together, we can, one thing that I want to show you here is that we can now look at a shift in conditions. So when E. coli shifts from a normal lab environment, which is what I'm showing you here, growth on M9 glucose medium, now we're going to shift it to a rich in medium where we have amino acids, okay? And a lot of things are going to happen. First of all, it's going to be sensed by a two-component system in E. coli. That's going to lead to the expression of many genes, which you'll see here in a minute as these change colors and sizes. That leads then to a shift in metabolism, which is going to have a large effect, all of which will then lead to the ribosomes being upregulated, right? The cells become longer and fatter. You can see the changes here. And then finally, what even starts to happen as E. coli grows in this very rich condition, it starts to parallel process its DNA replication, right? So that now it's producing DNA not only for its children, but for its grandchildren, and so forth. So there's a lot of complexity that you can capture all simultaneously if you're able to do this kind of modeling. And what we wanted to do is then take that and use it to evaluate these data. And I think we're in a society right now where data is kind of the dominating force. It's a data-centric approach to science, and I think that's great in a lot of ways. But it's also interesting just to remember that it hasn't always been this way. So one contemporary of Crick's, Sir Arthur Eddington, actually made this great point where he said, I hope I won't shock the experimental physicists too much if I add that it's also a good rule not to put too much confidence in the observational results that are put forward until they have been confirmed by theory, right? So we usually think about data as being the driver, right? If the theory doesn't match the data, then we get really, that's a problem. And it's true to some extent, right? But you also need to think about the other way around. As we start considering our data, right, it's important to realize that it's actually not all going to be correct. And Crick himself was a big contributor, or the big believer in this, right? He said that if he were forced to take all of the data explicitly into account, he would never have been able to accept the double helical hypothesis, right? Because not all of that data fit that hypothesis. They had to selectively ignore some of those data because they didn't fit with the theory that actually proved to be correct. So his point is that data can not only be wrong, but they can also be misleading. And I think we all know that, but it's important to say it explicitly. What you really need is not for the data to come forward or the theory, but really, in the end, we're going to need both, right? We're going to need both interacting together. We're essentially calling for people to do exactly what Delbruck said, except for now to do it in a larger, because the problems we're tackling are more complex, right, or rendered in more complexity, we have to use more complex approaches to be able to do that. Okay, so going back to how consistent these data are, what we did was we took these millions of data points, heterogeneous from hundreds, thousands of labs across the world, totally independently generated from each other. We took all of those things and wanted to see how self-consistent they are. So we're not testing the model in this case, right? Granted, the model is far from perfect, I get that. But what we're really trying to do is test the data against itself, using the theory to try and confirm the data. And so what we decided to do is use 3 different kind of metrics. The first 2 are kind of subjective, and the third is more objective. And I'm going to kind of fly through some of this just because I want to show you some of the other things that we found. But the criteria that we chose were, first of all, can we encapsulate this data mathematically? I'll show you what that means in a second. Second of all, when we ran simulations, did they make intuitive sense? Like, do they match kind of what known behaviors would look like? And then the third thing is when we go in and bring in independent validation data sets, do these data really line up with each other? And we tested it in the place where this model has kind of been built, right? The half that we consider in our model is the central dogma type processes, metabolism, and growth and replication. So looking at central dogma first, as I talked about in that first animation, we can write out equations, ordinary differential equations that govern all the RNAs, and their expression, all the proteins in their expression, right? So in total, that's going to be something like 10,000 equations, right? And this was the basis for us to start building this simulation. So what I want to impress upon your mind is that every single aspect of both of these equations actually has data that's associated with it, right? So you can bring all of these different things together, and you can link them explicitly in these equations. Virtually everything in this case is, I'd say the clear majority in this case is specified, which is really important to know. This is something unique to E. coli, right? We don't have this for really any other organism. But importantly, it's conceivable to generate information like this for every organism. So we can take those things, right, and then we can run our simulations. And in our case, what we're checking to see is if the behaviors that we get out are something that we would expect. So what we would expect is kind of exponential production of different proteins depending on their copy number. And you can kind of see here, there's a little bit, our simulations are stochastic, so there's always a little bit of variability. But you can see that by and large, these things grow out and double over time for this one cell simulation that we ran. You can also see that this kind of burst-like mRNA production that is characteristic of bacterial cells because of the low copy numbers of mRNA. So this all checked out. And then what we can do is start doing cross-validation between different sets. And the first thing we can do is stuff that you would be maybe more familiar with in bioinformatics, right? So taking microarrays and saying, okay, if we notice any gene shift in one microarray and somebody else has done a similar experiment, do those shifts match up, right? Or if we take what's been found in microarrays and RNA-seq and we compare that to what people established in the literature in the last several decades, does that match up? And it does, but this is kind of what I would say the bioinformatics method of doing these things. It can't really get you across these major heterogeneous data sets. For that, what you want to do is take those equations, right, the equations that I showed you, and then start leaving data out and seeing how well the rest of the data produces a prediction that you can then take with an independent set. So I'm just going to show you one here. We have more. But the key one that I'll show you is just taking the proteome, okay? So we took those equations, we filled them out, we just left out the proteome, and then we just plotted a graph where you can see, okay, here's what the equations would say based on all of the other data that the proteome should look like, and here's what the proteome actually looks like, okay? And so what you can see, I think it's pretty striking. Like obviously at those low copy numbers, there's more variability, but this is a very striking correlation, right? Clearly, these things interact very well. And in cases, like some of these cases that were extreme outliers, we actually went back and we tested one of the data types that we thought would be the least reliable, which is the half-life of the protein, which is usually just estimated from the n-end rule. And we measured those quantitatively, and in many cases, so 6 cases, that actually pushed them closer towards the line, right? So I think there is a very exciting and consistent story that's been being represented in central dogma type data. So then we can look at the metabolic network, right? And we can do a similar thing, right? For all of the metabolites, the small molecules, it's the same kind of idea. We use a different approach, a little bit, a slightly different modeling approach, but same idea, right? Again, what you're doing here is getting all of these parameters, right? Data types, and we're lining them up, bringing them together in terms of these equations. And once again, you can run simulations. Like in this case, we're running a shift. It's the same shift I showed you in this in the video, right, where you add amino acids, just like you would expect. The metabolic network shuts down production of the amino acids, right, as you see here. So simulations that match what you would think is kind of a natural thing that makes sense. And once again, we can apply this directly to, direct validation of the parameters. And what you see here is that we can apply all of these kinetic parameters, which actually, I spent personally a summer going through 1,200 papers and evaluating 8,000 more, just getting those parameters from each paper. Turns out that those really can be implemented mostly simultaneously, with some exceptions. The cell simulation will still live, right, and it will grow in a normal way, and I find that to be at least incredibly surprising. We can also then look at independent data such as fluxomics, where people have measured certain fluxes through enzymes. And again, they only had something like 20 points here that we could compare to, but a very nice correlation with a couple of outliers. So here now, it gives us kind of a platform to think, okay, well, if you have something that's very different, what's the story there? What does that mean? So it kind of pushes you in a new direction as well. The final, and I'm kind of purposely going through these validations a little bit quickly, so we can go to the next stuff. But the final thing that I want to talk about is looking at growth and replication. So again, what that does is it takes all of the molecular level measurements and it connects it to the cell and physiological measurements, right? Bringing those data together so that now you're explicitly linking, right? You've got a link from genotype to cellular components. Now you have a link from cellular components up to the overall physiology. And so once again, you can populate those things with a vast literature and measurement, which is great, and generate simulations like the one that I showed here, right, which capture this physiology. Now, again, we can compare this to a number of physiological measurements that have been made in 3 different environments, which is kind of where we focused our modeling, and they match very well. And just this idea that the same model, the same data, everything is all producing all of these graphs that you've seen. I want you to just keep that in mind, right? We didn't just take the one equation, the one set of equations. They are all together in this one framework. The last thing I wanted to show, just because I know Suk-Jun is here, right, is that also when we run these simulations, they match models that have been identified over the last several years, right? In particular, our model matches the adder replication phenomenon that Suk-Jun was a part of identifying and characterizing so well. And in fact, this is one of his great movies where it shows that growth. So taking all of those things together, right, it was actually funny. I wouldn't say disappointing per se, but when we first started doing this, I was thinking we were going to write a scorching expose on how the data weren't consistent with each other. But actually, they are strikingly self-consistent, which I think is really great. So then we started looking to say, OK, what do we get collectively out of this data that no one person would have gotten out of their lab doing their own set of time? And to do that, I want to tell you two things. One is, we have kind of a misconception in I'd say, the biological modeling field that I think does a lot of damage. So I'm just going to call it out specifically, which is there was a story told by Enrico Fermi where he said, I remember my friend Johnny von Neumann used to say, with 4 parameters, I can fit an elephant, and with 5, I can make him wiggle his trunk. Okay? So his idea was that if you went over 4 or 5 parameters, you were probably overfitting your system, right? So in our case, our model has something like 20,000 parameters. Okay? Importantly, almost all of those parameters, are, I think it's like 0.25% of the parameters that are not fit, right? Or that are fit. So everything else is fixed from the data. Like by and large, we try not to change anything that we get from the data. The cases where we do fit are related specifically to just making sure that a cell can grow and divide in a given growth rate. Okay, so those are what we change. And happy to discuss any of those details. But you can imagine, right, if you come from the von Neumann school, right, you might think, oh, well, how on earth can they have a model of 20,000 parameters that doesn't just completely overfit everything, right? So that's what I want to hit back on a little bit, right? So this is a very popular story if you haven't already heard it. And in fact, some physicists went through and published a paper where they actually did, with only 4 parameters, they were able to make this, right? And actually, if I could show you with the 5th parameter, they can actually get the trunk to shift a little bit. So they showed this, von Neumann was right. In fact, you can fit an elephant with 4 parameters. It seems stupid and obvious, but this is an elephant, okay? So there's absolutely no connection between their ability to make this very simplified abstraction, right, and an actual elephant. If you just think about this, the dynamics, just even that beautiful skin alone, right, would be impossible. I think just to make it crystal clear, and I think maybe the better analogy that I want to bring up is the idea of a massively complicated circuit board. This thing was designed with presumably thousands or tens of thousands of parameters, right, in a special software designed exactly for doing that. Now, if I were to somehow happen upon one of these in the wild, let's say, and want to reverse engineer it, okay, and my friend said to me, well, you can only use 4 parameters or else you're overfitting, right? I would think that is completely nuts. It makes absolutely no sense. So when you're thinking about complexity, whether it's a circuit board or a cell, what you really want to do is not over or underfit, but find the number of parameters that's consistent with the problem that you are trying to solve. So just getting back at that a little bit, I don't know. It's my soapbox today. So in that spirit, though, one of the questions that you want to ask, right, to get around this problem of maybe I'm overfitting or maybe these parameters are not a unique set. So one thing that we did is we went in and we said, OK, for every data type of the same type, what we're going to do is take those parameters and we're just going to scramble them around, right? So we're just going to assume maybe the exact values of these parameters don't matter, right? Maybe it's not going to have the same impact. So what we did is we generated 1,000 simulations in which the parameter values were scrambled, okay, from similar data types. So like protein half-lives were scrambled with protein half-lives, or like, you get the idea. So again, something I would not have expected, but it turned out to be, right, of those 1,000 simulations. So here, you're looking at a SBLOM graph, right, where you can see here's the division time histogram. Everything in blue is the simulation, is one of those scrambled simulations. And this red bar is the actual correct, all of the parameters came just exactly as they came from the literature. Okay, and what I hope you see is that in all of these cases, okay, the doubling time is much longer, right, than this one solitary simulation that has all of the parameters the way that they were intended from the publications. In the same way, you look at the proteome, and again, the correlation with the known data that I showed you before goes way down, except for this one lone simulation that has the data the way that it was originally inputted in the model. One thing that's kind of fascinating, if you compare to the fluxes, the metabolic fluxes, you don't actually see that connection, right? Which is partly because that part of the network is not as well specified, partly because the comparison that we make is to only 20 fluxes, and partly probably for reasons we don't understand. But I want you to see, especially in these two-dimensional plots, that there is no connection between this relatively, contained spread of these scrambled simulations and this one simulation that really represents everything we know and clearly is much more predictive of real behaviors and data. And the final thing I want to say about that is these are just the simulations which could actually divide at all. So for most of this, or for a third of the scramblings, they couldn't grow in a way, they wouldn't simulate in a way that would support growth, right? So what's amazing to me about this, taking this data set together, is that what we have here is a remarkably self-consistent and somewhat unique, at least given these simulations, somewhat unique ability to capture all of the data and the growth phenotypes, et cetera, that go with it in one unified whole. The second thing that came out of the data that we're still kind of breaking our heads on a little bit has to do with a really interesting phenomenon. So here is a certain protein, RNB protein, okay, and it increases in number over time, and then the cell divides, and it increases in number over time again, cell divides, et cetera. Okay, and here's the mRNA that produces that. And this is what we were expecting to see in most of the genes, like expression that looks just like that. That's common, appreciated way that things work in bacterial cells. But here is an expression that we never expected to see and were very surprised by. Okay? And what it is subgenerational expression. So what you have here is a gene that is actually only expressed, in this case, once over 6 or 7 generations, right? It's only expressed once over 6 or 7 generations, and when it's expressed, there's a massive burst, increase in the number of proteins. And then that increase it tapers out, but not really by decay, because those are stable proteins. Instead, over several division cycles, it just dilutes out of the population. Okay? So it's just a bizarre, at least seemed to me like a very anomalous expression, until we generated a histogram, and we found this is now the frequency of observing at least one transcript per generation. Okay? 100% is this red type, which you'd expect to see. If you look, almost 2/3 of the genes in E. coli growing in this condition fall in that category, where they're not being expressed every cell cycle. Okay? And the reason why we can be fairly confident about this is that the cell is constrained by how much space it can hold and how much it needs to generate to be able to reproduce itself, right? And the fact is, it can't contain all of this, right? And so we know, I'd say I feel very strongly confident that this is actually happening in individual cells, where a majority of the genes are being expressed less than once per generation. So going into that a little more, are there gene functions that we would find that are specific? Well, one thing that's interesting, right, if you look at the essential genes, okay, most of the essential genes are in that expressed every generation. That's good, right? That makes sense. Similarly, though, what I thought was very surprising is that virtually all, like a large majority of the antibiotic resistance genes, for example, are in this sub-generational expression group, which kind of raises this interesting idea, you know, as we think about how persistence and resistance actually plays out in a colony of E. coli. The way that I think about this, you know, if you think about us as a big colony of E. coli, somebody's about to wipe us out with antibiotics, right? So maybe just randomly, one of us is going to express that big burst, right? Maybe it's Bossum, right? He has this big burst of antibiotic resistance, right? E. coli doesn't really care if everybody dies except for Bossum, because Bossum can replenish the whole community, right? And so what happens is, in this case, in a very deterministic but still with a big element of randomness way, right? Antibiotics come in, Bossum survives, he grows out, he replenishes all of us, right? And now that's diluted out. So somebody else, maybe it's going to be Amy. She has the next burst. She is now the savior of the colony, right? And so in that way, E. coli is able to grow and grow in a very strong kind of the overall community is able to grow and repopulate, but it's always hedged its bets for certain eventualities by having this subgenerational expression. So I find it to be a very conceptually provocative idea, and we're very excited about it. So another interesting thing is that metabolism is more evenly distributed, which made between these groups, and this yellow group, by the way, is we never saw it expressed in the simulation, so that's also a really interesting functional area. So then we started wondering, well, how can cells compensate for the fact that they might have an enzyme they actually need, right, and it's not present for certain periods of time, like a metabolic enzyme? One answer to that is that this histogram that I showed you is environment dependent. Okay, so under some conditions, right, so for example, the lac operon, famously, right, would end up being in the blue in the condition that we simulated, but in another condition, like the condition with lactose, it would be upregulated, and so it would be in the red. And so we went and looked at all of the many thousands of RNA, seq, and microarray data sets to look at all of the different possible expression outcomes, and what we found is even then, okay, there are 330 genes that, strictly speaking, you always see in every single cell cycle. But there are over 1,000 that are always, as far as anything we've measured, they are always subgenerationally expressed. And that's pretty fascinating, right? Then there's plenty of motion between the groups, but this idea that there are some that are just programmed to be subgenerational, again, goes into this idea of antibiotic resistance or some other functions I could tell you about after. So that's one idea. Maybe it's just that some of them are only required in those conditions, right? Another idea is that maybe the proteins are just stable enough that even though transcription doesn't happen every cell cycle, you always have at least a few proteins in there doing their work. But another thing that came up we found to be more surprising. And so here you're looking at the menE gene, which is an important gene related to production of menaquinone. And what you see if you look at one of these simulations, okay, it's being transcribed up here. Got these counts of RNA. They're very low, right? And it produces a monomer. It actually works as a tetramer. And so you should see here, as this goes through, kind of it varies kind of hither and yon, but there are occasions, and here we're highlighting a particularly long one, in which there is no tetramer, okay, and as a result, the menaquinone is depleted over time. Okay? What's amazing is because the time constants which characterize metabolism are so rapid, once that tetramer shows back up, right, the pool can be replenished really quickly. So what happens is, I kind of thought of the proteins as buffering out, but it's very interesting to think, just considering this cell now as a whole, that metabolism itself is another redundant buffer that can also prevent the cell from having a deeply negative effect, can compensate for this subgenerational expression in these kind of rare events when you have a true depletion. Okay, so altogether what I hope I've shown you, right, is that we've been able to generate these simulations and hopefully also get some new insights. Certainly for me, they've been new insights. So what are the conclusions from this work? I think there are a few things. The first one is, again, like I said, I was hoping for a big expose. That turns out not to be the case, right? At least in this case, what I've shown you is a set of data, millions of data points, right, that led to these 20,000 parameters that is remarkably self-consistent, much more than I would have expected, right? And not only self-consistent, but as you saw with this kind of most recent thing where we did these scrambled simulations, even fairly unique in its ability to generate like real viable simulations and also to account for all of the data. So that's good news, right? We can breathe a sigh of relief as far as that goes. I think as a community, the E. coli community, I think, should be happy with what they've achieved in that way, right? It's a very self-consistent set. The second thing is just going back to this theme that I brought up with the modeling, right? It's not, sometimes we think of data as being in charge, right? Some theorists, just do their theory without ever thinking about the data, right? But really, what we need now more than ever, as we study more and more complex systems, and this is just the beginning, we need approaches that can do what this is doing, right? To take these data and simultaneously evaluate themselves against each other. And the only way to do that is with some kind of a complex theory. So that's what we need, right? Bioinformatics is not going to be enough. Correlation does not imply causation. What fewer of you maybe think about is the fact that causation doesn't even imply correlation, which I'd be happy to demonstrate to you on a whiteboard. But we've got to go beyond those correlations, and we have to get at the mechanistic fundamentals. Okay, third of all is not really a conclusion, but a question, right? Does this work actually represent a complete solution of any kind, right? I think most of us already know, and I would agree with you what the answer to that is, but let's just take that apart for a second. Okay, does this work represent a solution of some kind? Looking around online, right, some people, just the way people define solutions, like one thing that a solution should do is that it should account for the data, right, which I think we can say is being done here. And another thing is that it should provide some novel insight, which I think, again, we do pretty well on that account. So we could consider it a solution of some kind. Now, if we think about a complete solution, we've got to go back to Crick and what he actually meant, right? And here he says, by complete, one means complete in the intellectual sense, implying that nothing appears to remain which further experiment could not easily explain using well-established fact and ideas. Okay. Obviously, by that metric, this work falls far short, I would say. But I think the concept in mathematics of general and particular solutions can be very instructive here, right? Because a general solution is typically one that takes account for the entire kind of system, almost irrespective of parameters, whereas a particular solution is usually, specific to one case. And I think we could probably make some kind of a case that this is the beginnings of a particular solution, right? It accounts for a few environments, certainly not all of them. It accounts for a large portion of the genes, but not all of them, right? But the key thing to know from this is the reason we have these general and particular solutions is that the particular solutions are usually a springboard, right? A key part of formulating the general solution. And so my real hope is that as we keep moving forward, we'll be able to identify enough in the particular solution that we can kind of reinvigorate Crick's challenge and hopefully as a community start moving together towards the general. Okay, so with that, I'd just like to thank the center and thank my lab in particular for the great work they've done, and thank you all for your attention.
Markus, I was hoping to ask a question that links three things you said through your talk. First, the inability to come forward with the scathing criticism of replicability. The subgeneration.
Wait, sorry, say it again.
Your disappointment that you couldn't come up with the scathing
right.
commentary.
Yeah.
The subgenerational expression and the general and particular solution. I'm wondering whether having so much insight from 1,200 different directions has given you insights into why replicability seems more apparent. For example, do people think that they're holding more of the world constant and getting a more general solution and then see somebody else's data as not fitting, but in fact, they have 1,193 wiggly parts that change the whole solution space or something like that? What have you learned from that?
So that's a great question, and in fact, what this study focuses on is more like cross-consistency than reproducibility. One thing that I can tell you, having read like 1,200 papers looking for parameters, that is a little disappointing, right? It's extremely, so there's definitely variability in the kinetic parameters that people report. It's more disappointing, actually, to see people not willing to stick their neck out, right? So you'll see several papers from different groups where the first person who measured it measures a 5.1. The next group that measures it measures a 5.1. And the next group that measures it measures a 5.1. That's extremely difficult for me to believe, right, given what I see with all of these other parameters. So that is, if I were to say one disappointment, it would probably be that. Like, it seems ridiculous and maybe a little dishonest if you know somebody else's value to kind of presumably, I don't want to put any actions that they didn't actually do, but to me that seems pretty suspicious and not that great. The second one was subgenerational. So I'm a big believer that that there are many things that are going to come together, you know, from the whole set that won't come from any one point. And that's one thing I actually love. I love the idea that somebody from the 70s generated some data and that data finds new relevance today because we can put it in this big container. And my hope is, you know, I tell people this sometimes to a brain crowd, you know the old chestnut about people using only 10% of their brain cells. We all know that's not true. But I think we can agree that they use several orders of magnitude less of their high throughput data. I think that's probably, we're pretty solid ground there. So, so what I hope in the end is that people will generate a big data set, and then they'll generate a simulation of what that data set would look like, and then take all of the stuff that's the same and just kind of ignore it, because it means that we kind of would have gotten that part. It'll enable them then to focus, here's everything that we wouldn't have guessed, right? And focus on that. Sometimes reporters have asked me, like, well, does this mean the end of experiments, right? And I always say, no, it doesn't mean that, but I think there is a reasonable, actionable goal, which is what if we had the goal of no experiment wasted, right? Like every experiment actually gives us a new insight. I think that's something really to fight for that we could do using an approach like this. Over here.
So you had showed a slide that had a bunch of biochemical pathways and showing how your models were built on the backbone of some of those pathways. And there was this golden era of biochemistry that probably ended in the 70s when all these pathways in the years and decades leading up to them were elaborated. But that era kind of came crashing down, not because everything that could be known about enzyme kinetics and particular pathways and where they intersect had been defined, but because molecular biology became easy and those experiments were difficult and people just stopped working on them. But then there have been subsequent discoveries made through like cancer genome sequencing that some of these enzymes don't do what we thought they did, like isocitrate dehydrogenase, fumarate hydratase, two Krebs cycle enzymes that turn out to have very important roles in nucleotide metabolism, epigenetics. So if you have places where your data doesn't meet expectations, can that point to areas where the knowledge is incomplete and then make experimental predictions about what activities must exist but have not yet been defined and then go out and identify them?
Yeah. That's what you've mentioned there is like one of my deepest hopes, right? That eventually what will happen is there's kind of a closed loop discovery process in which big data set is generated, model comes up with something, there's discrepancies, which are sometimes bugs, definitely, but also enriched for new insight. Then there could even be like a component, although I've hardly developed this even in my head, right? A component where the model could potentially learn from itself, right? So try to help you focus on what the experiment of the most predicted insight would be to follow up on that. And I think exactly that, right? You could get at this idea of like, well, there's some function in here we haven't found, right? Where is it? And our lab has done that on a very small scale, but I'd really like to see it being done in an automated way. I think this really, the time has never been better to at least try to do some of these crazy things, right? And push towards a future where we're like automating discovery and where we're not just like discovering medicines, but designing them, right, in really exciting ways. I see that as years in the future, but a very exciting potential future. Yeah.
Yeah, nice talk.
Thanks.
First, I wanted to say when you had Eddington's quote up there, it made me think of Groucho Marx, who may have said it best. Who are you going to believe, me or your lying eyes?
Yeah, that's right. That's right.
So in terms of the complete picture, I don't know exactly what E. coli biologists have been doing since the 70s, but I'm guessing most of this is in the lab, right? Mm-hmm. And so, so quickly, first of all, the subgenerational expression thing, of course, runs up against the problems with group level selection, which I'm sure you're familiar with. Could be, but there are only very special circumstances where group level selection can retain something like that. And so my question to you is, how much of your current model that isn't really looking at E. coli in the wild is like taking that beautiful elephant and making cell culture from it, and then looking at cell culture? And another way of putting that in the form of a question is, if we looked at all of the E. coli in our respective guts this morning, I'm going to guess that way more of those genes would be expressed, not subgenerationally, but all the time in that arms race with fungi and phage.
Totally. And in fact, okay, so a few things. One is I would say group selection, in this case, by exactly what you talked about. In the wild, it's very, very different. Some of my favorite theoretical work in this area comes from Uri Alan, who looked at basically how a group of cells might optimize in changing environments, right? Like if you have an environment that is changing very regularly, E. coli is totally doing that, right? Through the oral fecal route, right? Just its wild kind of habitat is not constant at all. So in that case, you would definitely expect to have a more complex optimization. So in a lab, yes, you can run things out to get something totally optimized to grow on glucose or on some other component. But when it's in a totally changing environment, some of that stuff really does change, right? Where now it is kind of like we just heard today in this previous talk, although in a totally different scale, right? Rare events matter. They matter more and they matter to the whole population. If you're in a condition where you're periodically experiencing antibiotics, it's going to matter to have that group around. So the second thing was reducing it down to cell culture. I totally agree. And in fact, we have a lot of in-house knowledge these days about at least great hints about what many of the unknown genes in E. coli do, kind of in part motivated by some of the stuff that we've done. And a lot of it has to do with kind of behaviors that you could easily imagine being part of his life cycle that we just haven't tried at all, or until now, we haven't tried in the lab.
And just quick follow-up, is it possible to actually look just with natural like fecal specimens at that question of how many genes are being expressed regularly? And people
that's
have done
something
that?
you can do, and it's, well, interestingly, right, so this is just a lab thing, but if you go from a minimal medium to a rich medium, for example, it shuts down even more. Right? So, it can be counterintuitive, would be my only thought on that.
We're going to stop for our morning break now, and so
Okay.
all those with questions, please find Marcus over coffee. There'll be coffee and pastries right outside. We'll reconvene at 10:40 for our next session of morning speakers. Thank you.
Awesome. Thanks, everybody.